Good morning to everyone present here. I'm delighted to see you all. I am Sharanya Dumre Patil and I will be your host. It is with great privilege and pleasure that we extend a warm welcome on behalf of Amity University, Mumbai. Today's lecture will be delivered by the esteemed speaker, Dr. Amit Yadav. As we embark on the second week of our Biology of Tuberculosis lecture series, I am pleased to introduce the sixth lecture in the series. Today's exploration takes us into the world of cutting edge scientific advancements with the captivating topic of listening to the war cry of the immune system using a novel multiplexing technique. So far, the lectures have been delivered by Professor Dr. Sarman Singh, Dr. Deepak Kaushal sir, Dr. Savita Kulkarni ma'am, Dr. Anand Krishnan sir, and the most recent one was by Dr. Ved Prakash Dwivedi sir. This series is organized by Amity Institute of Biotechnology. In the complex interaction between tuberculosis and pathogen, the human system and human immune system, researchers are utilizing innovative tools to decode the nuance of this battle. The novel multiplexing technique, essentially listening to the immune system's response, offers insights into the intricate molecular dance within our bodies. Our distinguished speaker, a leading figure in his field, will illuminate the power of multiplexing techniques in unraveling the immune system's reaction to tuberculosis. These insights have potential implications for enhanced diagnostics and therapies. This lecture propels us into scientific exploration, peeling back the layers of understanding. With gratitude for your presence, let's divulge into the world of multiplexing techniques and their role in deciphering immune system's language in context of tuberculosis. That said, I would request Dr. Sashank Kamble sir, an assistant professor at Amity University, Mumbai, to introduce our today's speaker. Over to you, sir.
Yeah, thank you so much, Sharanya, for the introduction. So, Dr. Amit, I know him for since long. We used to play table tennis together when we were in PhD. So, Dr. Amit has obtained his BSc and MSc from Delhi University in botany. Then he did his PhD in from IGME, as I said earlier also. So then he joined the THST and he worked with a very big group, Dr. Kanuri Rao. Actually, is one of the very big scientists in the field of Indian science and typically in the field of tuberculosis research. His main focus at THSTI is to look at the post-translational modifications of the proteins and how that is regulated and how basically these PTMs affect the homeostasis and the cell fate, and whether this PTM memory is conserved and it can be passed on to the next generation. So these key questions he is addressing. Apart from that, he has also interest in big data in biomedicine, in high throughput data analysis, data visualization using computer science methods, as well as metabolic disorders, as well as social and biological networks. So he's a great person, very friendly person. So without any delay, I would like to invite him to start his inaugural talk.
Thank you, Dr. Shashank, and thank you for reminding and reminiscing the days we had at IGIB. And thank you, Sharanya, for the great introduction to the talk. Just a disclaimer before we begin. I am a computational biologist, not really a TB person, but as Shashank mentioned that when I joined THSPI, I was drafted into Dr. Kanuri Rao's group, where they were looking at how macrophages respond to TB. And with my interest in quantitative proteomics and PTM biology, I wanted to see if we can devise this technology to study temporal dynamics. So that was the idea behind starting this thing. So let us begin the journey and see how bacteria and the host talk. Yeah. So we are all aware that there's a great tug of war between the bacteria and the host. The macrophage shown on the left tries to induce some factors when it encounters a bacteria or any other kind of infection. Here we are talking in context of TB bacterium, S37RV, and it generates some proteins which are synthesized after it has seen an infection, right? So after it has seen an infection, it will translate some new proteins It could also be probably the ones that are already present, but we presume that the immune system after infection will produce something new. This something new is more important than what was already synthesized in the system, because that would be at a homeostatic level. But the new ones would actually partake in a conversation with the bacteria or the infectious agent. And then, depending on who wins this war or who is able to capture this dialogue more eloquently, will determine the fate of this interaction. That means the bacteria, if is able to hijack this communication, can actually fool the macrophage and start living in there. On the other hand, if the immune system is able to recognize the bacteria as foreign particles, and then can probably induce cytokine response. If it is not able to kill it, it will demolish itself using autophagy, and the antigen presentation and other kind of activities will take over. Cytokine storm will initiate, and other macrophages can come and start clearing the bacteria. So depending on who is on a better pedestal in this conversation will determine the fate. So for this, there have been several proteomic investigations, mostly in transcriptomics because it is easier to do. But in proteomics also, there have been several forays into this, how immune cells respond to infections. The challenge, however, is that we measure the whole proteome, where we are unable to segregate what has happened after the infection. So that was the goal of this study, or rather application of this technique that we were devising. So the outline of my talk is as follows. First, because proteomics is slightly complex technology, I would want to introduce to some basics of this. Then moving on to quantitation, because quantitation is not inherent in mass spec proteomics. Therefore, you need to know a bit about quantitation before we jump onto the higher order multiplexing, which is heteroplexing and boneplex, the techniques that we have devised for this study. And based on this, we'll talk about some application in MTV biology using immune system response. So we were trying to quantify the proteome temporally. So the first part, introduction to proteomics. And before we go to even introduction to proteomics, because many people are involved in doing genomics, transcriptomics, metabolomics also. So let us first understand this plethora of the techniques that are available to us and their role in understanding biology and the phenotype. So we know that genomics tells us what can happen, but with intervention from diet and environment, whether it is at organismal level or even at cellular level, it determines what is going to be our transcriptomics, or which tells us what is likely to happen, because not all genes will be transparent. From this transcriptomics, we get some proteins. Not all RNA will translate into protein. This is also well known that transcriptome and proteome do not have a one-to-one correlation. Therefore, studying proteins is much important. So it will tell us what is happening. Let me take a pointer first. Yeah. So proteomics tells us what is actually happening, because proteins are the workhorses of the cell. These are more closer to the phenotype than genomics and transcriptomics. And then metabolomics means the small molecules in the cell which are either assisted or used, or in the process, they are the substrates on which proteins also work, and they tell what has already happened. So for metabolic diseases, we tend to measure metabolome, but for other kinds of diseases, proteome is where the major action is. And this leads to the phenotype. So that's the importance of proteomics. So, this is the central dogma of the cell, where DNA makes mRNA, and with the help of tRNA, which makes proteins. One of the important things that is generally missed in this picture is that even if you want to make DNA from DNA or RNA from DNA, at every step of this central dogma, proteins are most important and crucial biomolecular players. And it will not be entirely wrong to say that this is a full cycle. Without proteins, you cannot even make proteins. So, but The challenge is that if you want to study proteins en masse, that means at high throughput level, looking at the systems level. So we are generally systems biologists. We tend to not look at things from very close, but from 30,000 feet, so that we get a bird's eye view of the processes that are going on. And from there on, we can zoom in on the important ones so that better therapeutics can be developed. So using mass spec, this is a challenge, but unfortunately, mass spec is the only technique that can deliver such a high throughput data. So if we talk about biofluids, there's a range of abundance difference from the lowest abundant to highest abundant proteins, which spans an order of 10 magnitude difference. That means 10 to the power 10 is the concentration difference between the highest abundant and the lowest abundant protein. There's no technique that can actually span this dynamic range. Then there's no equivalent of PCR that we can amplify what we want to study, like genomics. So therefore, you have to study whatever is present in whatever little amount it is present. Then alternate splice forms of a gene can create different forms of a protein. Therefore, not everything is well known in advance or completely predictable. And to top it all, you have more than 300 biological post-translational modifications, which we cannot directly deduce from a gene or an mRNA sequence. So these things make proteomics a great analytical challenge. So we have the answer in the form of mass spectrometry, which in simple terms is just a chemical weighing balance. So you try to measure molecules and atoms. So, but there's one caveat here. You need to have them as charged particles because mass spec otherwise cannot see them. So if I just take an example of a pen, if you can see this, if I just try to measure this using mass spectrometry, I won't be able to do it. But if I just add a proton to it, now it is a charged particle, we can measure this. So that means charge is an important factor. Otherwise, mass spec is blind to uncharged particles. So there are two techniques you may have heard of: MALDI, which is matrix-assisted laser desorption ionization, and ESI, electrospray ionization. These are called as soft ionization techniques because these were the techniques that could impart a charge to the proteins or peptides without breaking them, and because of which both were awarded Nobel Prize in chemistry. So in matrix, you generally co-crystallize your protein, rather peptides, not actually proteins, because proteins are larger molecules. So we first chop them up into small peptides using some enzyme like trypsin, and those separated proteins on gel, in case of multi, are then co-crystallized with a small aromatic volatile compound like cyanohydroxycinnamic acid or cyanohydroxysuccinic acid. So these, when are hit by a laser, generate, these are volatile, so they will generate a proton quickly and otherwise get evaporated. Excuse me. So from these protons, the peptide will take up the charge and get ionized so that they can be detected in the mass spec. On the other hand, in ESI, you have a liquid chromatography connected to this, in which you have a gold tip needle through which things are flowing. And because of which your analytes from this liquid can easily take up a charge. This gold tip needle tapers up at the end. So a droplet keeps forming here. Once the droplet gets enough size beyond it, which it cannot hold, this is at high temperature and pressure. So this will gain the charge from here and get ionized so that it can enter the mass spectrum. A mass spectrometer, this is a cartoonic representation of a mass spectrometer where we talked about the ionization source, which could be either MALDI or ESI. From here, the peptides will enter this vacuum chamber, which is electromagnetic in nature. So charged particles have a predictable path inside an electromagnetic field. We have studied this in our physics lessons. So smaller the mass to charge ratio, the faster it will be. Higher the mass to charge ratio, it will take some time to reach the mass detector. Sorry. So the mass analyzer actually exerts this separation power in the form of electromagnetic field. There are different types of mass analyzer. I'm oversimplifying this, but Just for your understanding, try to assume that if a particle or a peptide has smallest mass to charge ratio, it will hit the detector first. This is kind of a galvanometer, which detects impinging charges on this. And depending on how much type of ions it hits, some abundance related intensities are generated, which is called as the mass spectrum. So in a nutshell, when we talk about proteomics, we have a protein. So this is a simplified version. You will have actually thousands and thousands of proteins in one complex mixture. You digest it using trypsin, which is a site-specific cleavage enzyme, which will cut right after lysine and arginine, if not immediately followed by protein, and you will get some peptides. So these peptides, when enter the mass spec, will generate the peaks. There will be some noise, of course, which we will ignore, but the most intense peaks will come from the peptide, as shown in yellow here. So this is a fingerprint of a particular protein. But when we talk about liquid chromatography based mass spectrometry, it is not one protein from which all these peptides are originating. So therefore, we do not do peptide mass fingerprint there. This is only valid for MALDI, where we have a separated protein into the gel. We cut the gel piece and then feed it into the mass spec. So this would have been enough there, where these peaks in combination would be a unique fingerprint for a particular protein. But when we talk about liquid chromatography, we have several proteins at one go digested together and separated using an HPLC, which then enters the mass spec. So now this is called as tandem mass spectrometry. Earlier, we only talked about one mass analyzer where we get only peptide masses, intact peptide masses. But in tandem mass spectrometry, this is only the first step. So after MS1, where we get peptide masses, we select top intensity peptides for a second cycle, where two mass analyzers are put one after the other in tandem. So in the second mass analyzer, we have a fragmentation chamber called as CID cell, which is collision induced dissociation cell. So one type of ions are accumulated in that cell and energy is imparted. So now, and it is in presence of an argon or neon like gas, which will not react. So there is no chemical reaction, only physical interaction. So when they collide with each other, your CONH bond, the peptide bonds will break. And based on that, now you generate amino acid breakages, which is actually the sequencing information. Now you can actually sequence the peptide. So when the bond breaks between your CO and NH, you get B and Y ions. Y ions on this side, B ions on this side. Sometimes it can also break slightly here and there, but this is called as low energy CID. So we expect only B and Y type of ions. And based on that, it is completely predictable that if I knew the peptide sequence, I can calculate what masses will be formed. I have shown some masses in red here. So that means if there is a modification at T, let's say there is a phosphorylation at T. So instead of these masses will change by addition of a phospho group mass. That would be somewhere around 80. So I have not added that mass yet, just to depict that this would be some other modification as well. So if I know a theoretical spectrum, which I have predicted here, because we know the masses of all amino acids, and if my experimental spectrum comes here, we can try to match, and depending on the goodness of fit of this match, we can tell what peptide it was. So this is peptide level sequencing. So this is the whole process in a nutshell. We have a protein sample. We generate peptides using trypsin. Then we feed it into the mass spectrometer and we generate thousands and thousands of MSMS spectra. On the other hand, on the computer, we have a FASTA database where we take the complete proteome of the organism of interest, for example, in ammonia. We theoretically digest it using the same enzymatic rules that were used in the experiment. So we generate a peptide library. From this, we perform a peptide mass filter, which means although we may have millions and millions of peptides here, but we don't match each and every peptide with this data. Here, if I am looking at one spectrum, I have the parent mass. So for that parent mass, within some error, I pick all the peptides from this, which are called as candidate peptides. So for every single MSMS spectrum, this process is repeated. So if I have 1 million MSMS spectra, so this process will be repeated 1 million times. But for each single iteration, I'll have some candidate peptides which will likely match, one of which should likely match this MSMS spectrum. So for this, we generate your theoretical B and Y ions as depicted earlier. And based on, again, this match scorer, we can identify which peptide it was. So this is the whole process of proteome. Now, but this was only identification. How do we quantify? Mass spec in itself is not quantitative in nature. But as I also mentioned earlier, looking at the peak intensities, it can be used as a proxy for quantitation or abundance. So there's this technique called stable isotope labeling of amino acids in cell culture, or SciLab. We take light, medium, and heavy culture media in which we remove some essential amino acids. So which could be lysine or arginine. So when we remove these essential amino acids, we feed the normal state here, which is called as lysine 0, arginine 0, means there is no additional mass. That does not mean these are zero mass. These are not having any additional mass. But we incorporate some stable isotopes in the other one where we replace some carbon with carbon 13 or some arginine with the heavier version. So depending on which combination we can use up to 3 plex here. So this will be normal, which is called as 0, 0. This we generally call as medium, and this as heavy. If there are only 2 states, we call it as light and heavy. So depending on the signal of same kind of peptide in these 3 channels, light, medium, and heavy, we can, so this is in MS1 because this is complete peptide equilibrium. And we can predict the mass differences because we already know which lysines we have used the heavier or medium versions for. Once these are mixed in 1:1:1 ratio, their intensity differences should reflect their quantitative differences in there. So this is how quantitation is done at MS1 level. So looking at their MS1 peaks, which should be verified by MS2, whether these sequence identities are also the same. Once these 2 things are confirmed, we can use this for quantitation. People can also use only 2 states if they do not have a third level. They can use only 2 states, but they have to do this experiment multiple times, and they can still increase your multiplexing capacity. But it becomes more arduous and also not cost effective. There's another technique which is chemical labeling called as ITRAC, isobaric tags for relative and absolute quantitation, wherein if I have 4 different biological states, I can, after digestion, I can start labeling them with 4 different types of tags. So these tags have this composition where you have a reporter, a balancer, and a peptide. So the peptide, the same peptide from 4 different groups will be used. And so this is not selectively used. Everything will be labeled, but we'll come to it later. So the same peptide in different samples, 4 biological different samples, will be labeled separately in different vials. So in one, so the reporter and balancer mass is constant, but it will have unit mass difference between the 4 groups. So this is 14. The other one will have 15, 16, and 17. Reporter mass is what we see in the MSMS, but to keep the mass same, the balancer will be adjusted accordingly. So the total mass remains the same in all 4. And after labeling, ITRAC labeling, all of them will be pooled together and single MS will be done. So in single round of MS, you will see 1 peak. But when we go for MSMS, in the low mass region, we can segregate these reporter ions. The balancer is lost because it does not take up a charge. So we cannot see the balancer in mass spec. But reporter gets the positive charge. It is designed in such a way. And therefore, we are able to quantitate based on their intensities in MSMS. So the advantage is that in the same MSMS spectrum, we can do both identification as well as quantitation. I'll show this in a better way. So there are not just these 4 tags, there are also 8 plex ITRAC labels. So as I said, your reporter will keep changing by 1 unit mass difference, and your balancer is adjusted accordingly. And the same peptide from 4 different samples, when pooled together, will give you the quantitative value. Similarly, in 8 plex, we have the same total mass of 305 and unit mass difference between the 2. You might notice that 120 here is missing, and that is because phenylalanine has a peculiar ammonium ion released at this mass. Therefore, it will create challenges in quantitation. Therefore, it has been skipped, and the next one is 121. So using either 4-plex or 8-plex, we can perform higher order multiplex quantitation. Usually people use this for sometimes 4-plex or 8-plex or 6-plex also. I mean, 8-plex is your highest capacity. So this is in general the ITRAC workflow. You have samples which could be 4 or 8. You perform digestion, you perform labeling. So your all N-terminal get attached with this ITRAC label, and then you mix them together and separate by HPLC and perform mass spectrometry. In mass spectrometry, you will see that these ions are used for sequencing or identifying the peptide, like we told earlier. But in the low mass region, you will have all these quantitations added up from these 4 or 8 samples, depending on which one you are using. And once this is resolved, you can actually see. So this is resolved using computational methods. I'm just showing inset here, but this is evident directly in the spectrum also. You just have to zoom. So these masses will show up, and you can actually see which biological sample had what quantity of the same peptide. And in 8-plex, similarly, you will get 8 different quantitative values. So what is hyperplexing then? If we want to extend the capacity of multiplexing, there was this group at Harvard Medical School. So what they did, they combined MS1 based quantification, which was 3-plex, light, medium, and heavy. In light, medium, and heavy, you will see things like this, and you can quantify in 6-plex like this. So this is ITRAC, and this one is silent. But when they combine the two, I think I should just repeat this once again. So in MS1 quantification, this is SILAC based, like I told earlier. Your MS1 peptides will have a predictable mass difference between light, medium, and heavy, and this will tell you the quantitation. In MS based, which was like ITRAC or TNT, there is another label like ITRAC. You will get one peak, which will have all 6 labels together, and these 6 labels can be resolved in MS. So if we combine the two, you will have light, medium, and heavy, each with their own 6-plex of ITRAC. So now this becomes complicated because mass spec can only see them as 3 different peaks. And once you resolve this, then only you can go for these 6-plex quantitation. So how that team used this was, they used the light, medium, and heavy as replicates. So that means your light, medium, and heavy will not have any quantitative difference. Therefore, it became kind of a bit easier. So in a way, even though they were doing 18-plex, it was not really 18-plex. It was only 6-plex because you were only looking at these 6. These 3 were actually only used as replicates. So we decided, should we use this multiplexing technique and try to study temporal dynamics of immune secretor? So what we did, we developed a new technique using this as BONCAT. So there was a technique called BONCAT, which is bioorthogonal non-canonical amino acid tagging. It means instead of methionine, it is replaced with AHA, azidohomoalanine. So this has higher propensity to get incorporated. So you deplete the media from using, so methionine is removed from the media, you add AHA, and all the newly translated proteins will now have AHA. Using azide chemistry, this can be pulled or selectively enriched, and you will only study the newly translated protein. This is important in biological context for the reasons we have already mentioned. If you want to know what happened after some exposure or after some event or biological event, then you will need to study newly translated proteins. Otherwise, the whole proteome will have higher background. So you may have, let's say, 100 picogram of a protein, but 1 picogram of a newly translated protein will get masking effect and you will not be able to see it. So therefore, you need to selectively enrich. The other technique, SILAC and ITRAC, I have already explained. So they have been used together for different applications. Hyperplexing is the one that I explained here. So we combined all 3 to make BONCAT. Idea was that if we allow infections and in control, as well as different strains of MTB, different types of virulent strains of MTB, and we wanted to study 6 different time points. We did not want to use any of the labels as the replicates of each other. We will use actual replicates. So could we do 18-plex quantitation? So that was the idea. So with this, we thought that let us apply this technique to study the temporal quantitation of the immune secretome. And based on that, we devised this experiment. So we took macrophages, monocytes that were developing into macrophages, THP1. And so we devised 2 set of experiments because we could not go up to 36 plex. We thought let us do 2 experiments of 18 plex each. So in each experiment, we have a control, individual control. And the first one we infected with 37RA, which is an avirulent strain. The second one with RV, which is a virulent strain. Then we also used more aggressive strains, BND and JAL. One is a slow grower, the other one is hyper aggressive, but both of them have higher virulence than the RV strain. So we thought, how do we now study only the newly translated protein? So using SciLab labels, we differentiated between the strains, and using AHA, we only specifically tagged the newly translated ones. Now we wanted to see what is secreted out and only look at the supernatant. In another experiment, which was done by somebody else, they were also looking at the palette and the whole proteome, but in this study, we are only talking about the secretome. So this is the design of the experiment once again. So at time point 0, we started enriching with AHA and SciLab, the respective SciLab. And for both set 1 and set 2, I will just re-mention this. So set 1 is RA and RB, set 2 is BND and JAR. So for both set 1 and set 2, after 6 hours, whatever proteins are labeled with AHA will be read here. Similarly, in different sets, we'll only enrich the proteins, then 4 hours each after the first 6, because initially it takes some time for the infection. And after that, after 4 hours each. So in the specific time window, we enriched for newly translated protein. So we wanted to see the temporal dynamics only in that specific window, so that the other background proteome will not create issues, because we know secretome is really low abundance, so it is already quite challenging to study it. And therefore, if we have a background problem, it will not be visible what is happening inside the immune cells. Then using these 18-plex set of 2 sets of 18-plex experiment, we labeled each one with ITRAC, respective ITRAC. We used only 6-plex here because of the design of the experiment. Then we collect their supernatants, separate them using LC. Now this LC was done in 2 ways. We separated them in linear chromatography as well as non-linear gradient, because sometimes too many proteins accumulate at one place. So therefore, we use both linear and non-linear gradients, so that a separation in either one of the 2 will be better. And then we combine the data. So and for each of them, we had 2 replicates. So we combined all of this data together. And now you can imagine at 6 different time points, we have 3 different strains. In fact, one is control, 2 infection strains. So we were studying 18-plex in one single experiment and 18-plex in the other experiment. So we had 2 experiments of 18-plex each. So this generated somewhere around 2.5 million spectra, which was quite huge, and hundreds of GBs of raw data. So our next challenge was how to segregate these different tag, 18 different tag lib spectra separately, and be able to quantify it, because we were not using replicates. So there was no tool that was helping us do this. So we used MuxFont, and we had to develop our own tools, one of which was Fontwiz for spectra level quantitation, and then HyperQuant, which used identification from MuxFont and quantitation from Fontwiz, so that it can combine the data. So just to explain why this was a challenge, any tool, if I tell that I'm doing MS1 quantitation, it will add up all these time points as one, and I will only see the accumulated intensities from these 6 time points as one quantitation value. So it is possible that their different time points may be different, but the overall value may turn out to be same, and it will say there is no quantitative difference between the two. So therefore, it will be a challenge. So if I tell it is a MS1 label, everything will be clubbed from in each of these columns I'm showing here. But if I say it is MS2 label, then it will club in the other manner, and it will again be a challenge. So this is why we had to develop our own tools. Therefore, we developed this hypercount pipeline. I am not going into too much detail because, as I understand, there are not many computational biologists here, but just understand that for after matching each of their peptide spectral matches, we individually segregated these 18 different conditions using our tool that we had developed, mapped and clubbed them together so that we can have statistical calculations and perform the quantitation accurately. Otherwise, they would be clubbed together. So that was the idea. These were the tools that we had to develop for this purpose. So then there's this question that how do we know whether our pipeline is working correctly? So for benchmarking, what we did, we went back to the hyperplexing data, although their experiment was not exactly same as ours. But if we can tell our tool to analyze that data, we could use that as a way to validate our approach. And we could show that for whatever we could identify, our approach was doing slightly better than them, even in their own data. And we had quite good correlations in whether we look at light, medium, and heavy peptides, or if you look at the temporal ratios. So we had pretty good correlation. This told us that our approach is working fine. So when we talk about secretion, so the idea is when the newly translated proteins, which are already at quite low abundance, when they are secreted out, it is possible that the abundance would again be more lower. How do we measure what is happening? So in the two sets, we saw that the secretion is reduced with respect to the control. So the control is able to secrete more proteins rather than the ones that are infected. And when we combine these two in one coherent map, even then the controls have higher secretion rate. That means overall dampening of secretion is happening when the macrophages are infected by the PV bacterium. We checked if we are wrongly getting some proteins or whether they are really secreted. So from whatever is known in literature, we try to map using high throughput mapping techniques. We found that most of the proteins that we had found are either secreted or predicted to be secreted or membrane associated. And very few are known as nuclear or which have no information. And most of these are either extracellular vesicles or directly secreted. So it appears that subcellular localization tells us that these are mostly secreted or predicted to be secreted. So we know that whatever we have found is fine in some sense. So we also try to map whether the two experiments have some differences, because in the first one we have clinical strains, laboratory strains, RA and RV. In the other one we have clinical strains. So we saw that there's definite down regulation of most of the secretome in both, and whatever is related to immune system response or cellular regulation, both are going down. So we saw that why not separate them by keywords and talk about the immune response specific to infection. So whatever processes we found, we again found that most of the proteins in infection are going down and down, although RA has slightly lesser effect than others. So when we talk about only the most common proteins in all the different conditions and all the different infections, we found that most of the proteins, again, as I was telling earlier also, are secreted and below their normal averages. So if you look at the gray zones, these are the zones where the secretion is normal. But if we look at below the gray zone, these are the ones which are statistically significant downregulation. And we can see that only in JAL, we have PPT1 and IL-1 receptor antagonist. So these are quite important for immune response, and these are completely being dampened by JAL taking control of it, because JAL is the most aggressive one of the four. So we created kind of a bird's eye view map of the whole process and showed which proteins in which different condition are getting up or down regulated. And we can see that in immune response, cytokine signaling, adaptive immune system, and all kinds of system that we studied, most of the proteins were going down, and we could ascribe it to many of the known immune processes. So we thought that, okay, this is a bird's eye view. Can we compile this into something more cohesive? Although it was quite challenging for us to do it for hundreds of proteins, we still selected the most relevant ones. And so you will have to look at this legend first. So this is RA in green. The downwards triangle represents down regulation. Circle represents no change in expression. Upward triangle represents up regulation. So this is same for all, and the four different colors are representing these four different species. So one of the most important things that I would want to highlight here is that if we look at MAP kinase, which is one of the most important for cytokine production, we see that except our JAL, most aggressive ones, most of the proteins everywhere are downregulated. Even when we talk about MTB inside the phagosome, most of the proteins are downregulated. There is some response in RA where it is able to induce some response, but then it is quickly controlled by the immune system. So that is why we only see some response in RA, but not too much response in the other strains, because other strains will mute the immune response so that it cannot call other macrophages or helper cells to kill the bacteria and contain the infection. So this is the crux of the whole technique application. These are some summary I have jotted down here, that Bonplex allows multiplex quantitative proteomics of host immune response to infection. This can be used in any other cell model that we want to use. And now it is possible to go up to much higher, because now TMT 18plex is there, which our tool can analyze, and Select 3plex, which means 54 different conditions in one single run of mass spec can be achieved, which will also remove the run-to-run variations that we generally see. We observed that majority of the proteins that we were looking at, they were only expressed in control or to some extent in RA infection, but not expressed in the other virulent strains, which hints that there is a huge suppression of the cellular function, and specifically newly translated proteins to be secreted out. Even if the immune system is trying to respond, it is being blocked so that these things don't get secreted out. Because once it gets secreted out, it can act as a flare mechanism for other macrophages or other immune cells to come to the site and contain the infection. One of the most important is IL-1RN, the receptor antagonist for IL-1, which inhibits the binding of both the chains alpha and beta to its receptor, and this reduces the inflammatory response. And in JAL, it is, which is the most aggressive one, it is the most prominent one. MAPK signaling also is quite important for signaling, and which leads to cytokine signaling, which again brings other immune cells here, which is also attenuated to a larger extent. Similarly, complement cascade, antigen processing, presentation pathways, they're all blocked. So there's a huge blockage of these immune response in other virulent states, except for RA to some extent, because RA is easily contained by the immune cells. So they have varying degrees of control based on their virulence. So these host factors are actually quite important, because now they can be explored further for the development of host-directed therapies. For example, if we know which host factor is either hijacked by the bacteria or helps the bacteria so that the immune system cannot see it. So all this bacteria requires is that it remains invisible to the immune system, right? So because it has to live inside. So if we know what glasses it is wearing to become invisible, we can actually attack that host factor or bring the levels down or up, as the case may be, so that this can be used as a host-directed therapy. Several host-directed therapies are being pursued, and this can actually add to the kitty lab. So since we were mostly method developers who were more interested in seeing how this can be used for different use cases, it is not only applicable to infectious diseases. It can be also used elsewhere because we are trying to ramp up the statistical quantitative values that we can study, which are more reliable than normal experiments. That was the major goal. So we had patented this technology, but now we can go up to 54-plex, not just 50. So thank you to the team who had actively participated in this study, and many people were involved. I'm just talking about it. So many contributors from my lab and my other collaborators who were doing the wet lab experiments. Although we were designing we were involved in the designing part collaboratively. So they are not here, but I should acknowledge them. Shilpa and Ajay did all the experiments on cell culture, while Mukul and Basira did the experiments on the mass spec. So thank you to my collaborators and my team, as well as the funding bodies who provided us with enough funds to do this work. Thank you.
Sharanya?
Now you can wake up and ask questions if you want.
Yes, it was a very good talk. So can I ask questions?
Yes, please.
Yeah, so basically when you take the supernet, the secretome, you basically take the supernatant upon
Yes.
infection with different strains of MTB. So my question to you is that how can you shortlist which of those proteins are specifically secreted in response to bacterial infection, and which of them are non-specific, a kind of
So
host response to
correct.
Yeah.
Right, that's a good question, and actually it is very important. Otherwise, you would not know what you're observing is just you're observing because it would be produced anyway, or is it specifically in response to this? So there are 2-3 factors here. And one is that for each of the set that we were using, we had a specific control embedded. So with control, you can actually normalize the values and you will know whether it is specifically secreted or not, because the amount will either go up or down. Here, most of the proteins were going down, so we could know, except for PPT1 and IL1R1 receptor antagonists. So this gave us the specificity that these are specifically done in response to the infection. And we had 4 different replicates of each. So that also gives us some statistical significance. And for different strains, we had different SILAC labels. So we could actually, even though we were only using one run of mass spec, although we did it 4 times, we were able to segregate each one of them by their mass pixels.
Okay. And since you're taking supernatant only those proteins will be detected which are secreted outside of the host.
Correct. That was the goal, because and also remember
those proteins which are produced or which are secreted in response to the bacterial infection, but which are still in the cytosol, not secreted outside. So they'll be escaped, right?
Yes. So there are several small caveats to this, but also remember these are not just secreted proteins. These are the proteins that are also synthesized after infection. Right. So these are newly translated and secreted. Right. So that would be the more appropriate term, that these are newly translated and secreted. So let's say if receptor antagonist is already present. So that means at some basal level, it is required for homeostatic maintenance, or let's say even for normal functioning. But only after infection, you will have newer synthesis of this antagonist. Right. Correct. And that will be pulled up only if they are secreted outside, because we are only taking the supernatant. It will be pulled up by our enrichment technique once they are newly synthesized, and after that they are secreted out, then only they will be taken. Right, great. So nice talk. And actually, I have been working on the bacterial proteins, bacterial secretome, MTB secretome, and its cross-talk with the host proteases for long. So we can definitely collaborate in this sense. Sure, I will have sure. With you. I have a lot of till now we have done in silico work, so that data is with me, and we can further collaborate for the subsequent experiments. So thank you, Dr. Amit, for such an insightful, wonderful talk, and I'm sure that the students
of AIB have been benefited a lot from this talk. He's a great person, a very good scientist, and as you know, we are very we are long friends. Okay, so thank you so much once
Thank
again
you.
for accepting my request to give a talk. Thank you so much. Yeah.